All right, so today we are going to have a webinar regarding to investigating dark matters in metagenomics using linked sequencing technology in metagenomes by Dr. Lu Zhang. So good morning, good evening, and good afternoon, everyone. Depends on what time zone that you are in. We have today's agenda to start with a quick introduction, and then we're going to jump right into the topic for the speech, and then we will have a quick Q&A. My name is Zhang, and I am the Senior Director for Global Business Development and Distributions of Universal Sequencing Technology, a biotech company based in Carlsbad, California, in the United States. Today, we have the privilege to have Dr. Lu Zhang, Associate Professor of Computer Science at Hong Kong Baptist University, with interdisciplinary background in genomics, statistics, and machine learning. The primary research interests for Dr. Lu Zhang are computational genomics, bioinformatics, and AI for genomics. His team works specifically on develop computational tools to support advanced sequencing technologies, develop foundation models for multi-omics data, and explore the application of large language models to address modern healthcare challenges. He was a postdoc fellow in the Department of Computer Science and Pathology at Stanford University. He earned his PhD in computer science from City University of Hong Kong and Master Philosophy degree from Li Kai-shing Faculty of Medicine at the University of Hong Kong. Without further ado, let's welcome Dr. Lu Zhang to give us a speech.
Okay. Okay. So first, thank you for Ruth's great introduction, and thank you for UST for the invitation to give me an opportunity to share our recent research advances in the field of metagenomic sequencing. So, so the topic of today's lecture is about actually, you see, advanced computational method to decipher the microbial dark matters using linked metagenomic sequencing. So our team developed a series of computational tools, which could be applied to both the linked arrays and like the hybrid metagenomic sequencing to perform the genome assembly and contact binding. So let's start my talk today. So there's some outlines. First, I will introduce some background. What is the metagenomic sequencing and what is linked array sequencing? I think most of you guys should be quite familiar with that. So the second part is our group's research work. So first two works about the metagenomic assembly on the, first work is a benchmarking work. We compare different tools on short arrays, metagenomic assembly, linked arrays, and also long arrays. And second work is also some work about specific on linked arrays, so metagenomic assembly. And we have some take-home message for you guys when you would like to adopt a linked arrays for your work. And third, fourth, and fifth, we will introduce three of our recent work. For Pangea, it's about the metagenome linked array assembly. And second work is depurifies after you got the assembled context, how to achieve a good quality metagenome assembled genomes. And last is the LRTKs or as toolkits, which supports the almost, I think, the popular linked array sequencing platform to do the data analytics. So, okay, so first, what is the microbes? The microbes can be found everywhere in environment or in the human body. We see that fresh water, seawater, soil, and even our gut, okay, our body or skins, anywhere. There are many different kind of microbes. So I think it is quite hot topic in recent years. So we would like to know, so how such kind of bacteria interact with example, human being, how to interact with host, how to affect our health, or for example, how to in the soil, how to affect the plant growth, etc. That is quite important, especially actually in the COVID period. So because in Hong Kong, so we use just the water to monitor the expand of the viruses, but that is viruses, but that is also use the same technology. So we call it like the metagenome sequencing. So here is in the left-hand side is how the microbiota so infect our human health. Actually, it has been approved to be associated with many different kind of disease, like liver disease, diabetes, or heart diseases. So or depression recently it recently work. So then that is quite important. But the problem is that because all the bacteria, they live in a ecosystem, stable ecosystem. Actually, so most of the time it's quite difficult to isolate the individual bacteria and culture it in the lab because we do not and fully understand the requirement of the living of the bacteria. So whether the temperature, whether it's a wet or dry environment, so what kind of food it would like to eat. We have very little knowledge about that. That is quite difficult to understand that. So actually, but our aim for the metagenomic sequencing is that we want to understand the composition of the bacteria in the samples. So when we would like to know the composition, we should first know their genomes, right? So we know the genomes, we can annotate them, or we can compare the genome to reference databases. We know what kind of microbes in the data, right? So if we would like to do that, we need a sequencing, right? We need to sequence this the genomic sequences of such kind of bacteria. So here is a brief introduction of the three different kinds of commonly used sequencing technologies. The first one is short-read sequencing is commonly used currently. The input is just the DNA molecules. We just share the DNA into very short fragments, for example, 100 base pair. So after that, we got the short-read paired and sequences. Okay, the advantage of short-read sequencing is actually it's quite accurate, as we know. But the problem is that the sequence length is quite short. That means it cannot cover the, for example, repetitive sequences in the microbial genome or in the human genome. So the third one is a long-read sequencing. It's short-read, for short-read sequencing, long-read sequencing. Long-read sequencing, actually, they commonly use the single molecule sequencings. So their rate is quite long. But the problem is that commonly, the most of the mainstream technology, like the ONT, Oxford nanopore, like the BGI cyclone, their sequencing error is quite high. Actually, ONT also is improved a little bit in recent years, but it cannot compete with the short-read sequencing. But of course, you can choose to use the hi-fi from PacBio to sequence the macro. But problem is that the hi-fi, they need a large amount of input DNA, and also price is a little bit high. Actually, it's not appropriate to be applied to large scale studies. So somewhere in the middle, we call it link-read sequencing. Link-read sequencing, we can also call it like, in our previous slide, we call it like read clouds or the long fragment sequencing. So that is technology, something between the short-read and long-read sequencing. First, it shared DNA to long fragment, but different from long-read sequencing. So shrink range sequencing does not sequencing the whole length of the long fragments. Alternatively, as I show in the right-hand side, you see that for each long range, for each long fragment, we just share it in the small pieces and perform the short range sequencing on that. And besides that, we attach the barcode for each short range. So we can just simply say that if we find that the short range with the same barcode, we can roughly regard them from the same long fragments. That means such kind of technology have the advantage of short range sequencing, high nucleotide accuracy, and also have the advantage of long range sequencing. So because we have a long fragment, we have barcode information, we can group short range. We know that which short range from which long fragments. So that is link rates, and link rates in DNA is quite low, and so it is cheaper, much cheaper than long range sequencing. So it is more appropriate to be applied to the large scale studies. Okay, so next is some platform, like popular platform, hospital popular platform our group have worked on. So first one actually is the the link raise technology. I think in 2016 and 2015 when I first joined my Stanford lab or work on that. So that is technology is a droplet based. If you have a gel, and so actually the long DNA fragment will be assigned to each droplet, and the droplets they will receive that for each genome gem. So for each droplet, they have the long fragment shared into small pieces, and we perform the stop rate sequencing of that. So after that, we have the barcode because we know whether the two short rays from the same droplet or not. Okay, so TX technology actually is good. It's good for human genome assembly actually. But they have two problems. The first problem is that because the technology limitations, for each droplet, commonly it is include more than one fragments. So for human genome, actually it is fine because for human genome, commonly the long fragment length is relatively long. The weighted fragment length can extend to 50 kilobase. So that means for each droplet maybe only have 10 fragments. 10 fragments means when you collected the same the short rays with same barcode, they could come from up to, for example, 10 fragments. For human genome assembly, I think that is fine because the sequence is quite different, and the fragment length is quite long. So you have sufficient rays come from the same fragment. But for the metagenome sequencing, the problem becomes severe because for linked range sequencing and microbial genomes, we cannot get such kind of long fragments. For metagenomic linked range sequencing, we only have like 5 kilobase to 20 kilobase length. That means more fragments will be squeezed in the same droplet. That will significantly increase the difficulties in genome assembly. Okay. Of course, the second drawback of this technology is the company discontinue to support the technology. So we have no opportunity currently to sequence or data using 10x linked range technology. So currently, alternatively, we have two kind of the alternative technology for linked range sequencing. One is from UST, Dr. Telsic, and one is from SLTFR from BGI. So actually, both of the two kind of technology, I think they like somewhat overcome the problem with 10x linked range sequencing technology because the barcode specificity is quite high. That means a majority of the short rays, if you find that they share the same barcode, that means they're from the same fragments. Okay, I think that is the metagenome assembly can benefit much from this advantages. Okay. So what does the data looks like? If we just use sequencing technologies to sequence your metagenome samples, you can probably have a like cartoon figure like this. You know, you can collect, you see the different color long fragments. You share the genome, for example, here bacterial 1, bacterial 2, into different long fragments, but you see the data is just like here, the short rays. But you see the short rays have same color, they are from the same bacteria. Okay, so then I will show more about the metagenome sequence as I just mentioned that it's quite difficult for most of the environment to isolate and encounter the single bacteria in the lab. So we will have the metagenomic sequencing technology. That means, so we got all the genomic DNA from all the bacteria in the sample, and after that, we perform the DNA sequencing of that. That means we got the row sequence rates. You see that the different color here not means barcode, it means the different bacteria. That means our input data is some row sequence rates. We know the rates could be from the different bacteria, but our aim is that how could we generate like the microbiome genomes here. So so then how to generate the microbiome genomes? Commonly we need the metagenome assemblies. That is because, you know, that maybe more than 90% of the bacteria, we don't know their genome. Okay, of course, we have a better understanding of the human gut microbiota, but for the other environment bacteria, we have very, very little knowledge about them. That is quite different from human genome, right? Human genome, we have reference genome. We even know currently we have T2T, we can use the reads to directly align to the reference genome. We know the variants, we know what happens in the genome. But for metagenome, for the bacteria, we do not have the high quality reference genome. That's proven us to perform the assumption-free techniques, algorithms. We call it metagenome assemblies. We just input the row sequencing rates, and we want to concatenate this rates to a longer fragment. We call it context, and even longer can be the circular genome of the bacteria. So how can we do that? So in computer science, commonly we use a graph-based algorithm to do that. Okay, so commonly we have used the overlap between the rays to just one by one concatenate rays. That is a very straightforward way. But problem is that if we do that, we generate graph, and the size of graph will be significantly increased during your sequence raising increase, right? Because each node in the graph is a raised. So in computer science, we start to use another data structure to solve this problem. Actually, we call it depth range graph. Depth range graph is based on K-mer. So K-mer is that we just shared short rays into the small fragment with size of K. And you see that here, for this example, we have K equal to 4, we have former. And for each adjacent former, they share the three the same nucleotides. So use this former, we can represent this rays. Okay, so at the beginning, so we will just use the K-mer as the nodes, and we use the overlap of a K-mer to be the edge. Then we have a Hamilton depth range graph. But Hamilton depth range graph, the problem that it's an NP-hard problem. It's quite difficult to be solved in a reasonable time. So the computer scientists decided to use the Euclidean depth range graph that for each edge represent K-mer. Then the problem becomes that whether we find a path in this graph that it can go past all the edges in the graph. If you go past all the edges in the graph, that means we go through all the rays. That is, we can concatenate such kind of short sequences to be a long sequences. So this is a definition of metagenome assembly. So actually, when we use the, for example, the range graph-based algorithm to perform the genome assembly, the problem that commonly we cannot get the complete genomes. We cannot get the circular genome of the bacteria. Then what is the next step? So we want to know, at least we want to know the marker gene or the essential genes of the bacteria to help us to perform the downstream functional analysis. So we have another steps, we call it contig binning. Contig binning, the rationale is that we want group the contigs from the same bacteria together. So how can we do that? Commonly, it's used two kind of the information. First about read depth. So read depth is especially important for if you have multiple samples. Read depth can reflect the abundance of the bacteria. You know that the different bacteria commonly have the different abundance. If we find that two contigs, their sequencing depth quite different. It's almost impossible they came from the same species, right? And also another one, TNF, is nucleotide frequency. That is reflect the sequence context of genome, because we have assumption behind is that we believe that the distribution of TNF for different bacteria, they should be different. So based on such kind of information, we can group the context into bins. The bins, so we will evaluate the bins, use some off-the-shelf software like the Checkam. After that, we know the completeness of this bin and the contamination of this bin. So such kind of bins will be separated in high quality, medium quality, and low quality. So we will treat them as also call them as metagenome assembled genomes, because that is genomes that is from metagenome assembly. So currently, I think most of the databases, they have include many such kind of MAGs from metagenome assemblies. Okay. All right. So next I will show some research work about the benchmarking metagenome assembly method. So see, this is our one of our work. We have through the simulation. We have several to mock community, ATCC and ZEMO. We also collected 4 different human gut micro samples, 2 samples, 7, 8 we sequenced by ourselves, and 9 and 10 we just downloaded from the previous work. So we will also try to use the different assembler. We evaluated actually 19 assemblers. Some of them, like the Metaspace, Mega is very popular short-range assembler. At that time, for linker assembler, we only have the Athena and the CloudSpace, also some like Canoe, Metafly, such kind of long-range assembler, and also the hybrid assembler. Hybrid assembler is a hybrid between the short-range and long-range. We use them to evaluate which kind of sequencing technology is there pros and cons, and the pros and cons for the different assemblers. Okay, so before that, let me introduce some evaluation matrix we commonly use to evaluate the genome assembly. First one, N50. So that is the weighted average contig length. We just sorted contigs based on their length, and we just aggregate the contigs. If we find that, for example, here we add contig A, the length is 8, the fourth contigs, we find the length exceeds the 50% of the total assembly length. Then the length of the first contigs, we will call it as N50, contig N50. And for contig NV50, the definition quite similar as N50, but only different is that when we calculate NV50, we need the reference genome. We need to cut the contigs. We find a misassembly inside of the contigs. So NV50 can be a good evaluator to rule out actually the misassemblies. And NGA50 is that we just currently NA50 use the total assembly length, but NGA50 we use the reference genome length. So for the NA50 and NGA50, we can only evaluate them if we have the reference genome, that means simulation data and the mock genome. We also, when we have the MAG, so we use a different, you see that very stringent complete contamination, and even the accuracy RNA, RNAs, 16S, 23S, 15S to evaluate the quality. We have separate them to near complete beans, high quality beans, and medium beans. Okay, this is our roughly result of that. So just in one sentence, for the continuity, of course, the long range assembly is the best because we have quite long sequence on that. The link race is something between, and the short race is the short is the smallest continuity in that. So besides of continuity, so more information we concern about is the MAG quality. So that is quite interesting. You see, when we refers to the high quality MAGs, we find that actually the long rates commonly have the smallest number of the high quality MAGs. Why? In some papers, they argue that that is because the low sequencing depth. Actually true. This is true. So commonly for long range sequencing is expensive. We cannot afford to, for each sample, we'll sequence 50x or 50 gigabytes on that. Commonly, we have 10 to 20 gigabytes. That could generate many circular genomes, that's true, but it will also lose some microbial genomes, especially for low abundance ones. Okay, and in this technology, we find the link arrays, surprisingly, right? It's got the highest number of high quality MAGs. I think because the first we sequence link arrays because they're from short arrays, this cheap, much cheaper. So that could make us to sequence more data, generate more data on that. We can also input more DNAs for link arrays sequencing technology to increase the library complexities, and also have the barcode and help us to make the context longer, much longer than short range assembly. That means they make us a higher chance to get the more genes in the same context in the MAGs that can significantly increase the completeness. Okay, so then we compare different sequencing techniques. We know that long range have the largest continuity, and link arrays have the largest number of MAGs. So the second question, we will just specifically investigate the metagenome assembly on link arrays. So this is a paper, roughly a little paper from my previous lab. We developed a computational assembler we named as Athena. It could specifically assign to the to design for text technology. Actually, this kind of thing is that the rationale quite simple. Is that when we have the we call it scaffold graphs, and if we align the rays to a scaffold graph, we find some paths is supported by the short rays with same barcode. We regard it, oh, they should be from the same bacteria. We should connect them. Okay, that is we just use the barcode information to concatenate the context. So based on such kind of method, so we just fully investigate such kind of technologies. So on the how does the scene apply to link array sequencing? Actually, link array sequencing is quite different from the short range and long range. We have more parameters on that. The quite familiar the parameters we know is C. Here is the total ray depth, not the same as short range and long range, right? But in link array sequencing, C equal to CR times CF. So CR is the here. It's a short range per long fragment. And CF is the physical fragment depth. Okay, because for such kind of the technology, CR is commonly quite small. That means it's quite small number of short reads share the same barcode because they're from the same fragment. But we will find that there are many different long fragments cover the same pieces of genome genome locations. So we have the CFCR. We also have a mu-F and mu-FL and W-mu-FL. That is fragment length. That is unweighted and weighted fragment length. And NFP is the number of fragment per partition. That means for each beads or for each droplet, how many fragments will be assigned. That means for the reads with the same barcodes, how many of them are from the same fragment. Okay, so this is just roughly, because the time issue, just roughly goes through the result here. This is from real data. Actually, we find if we sequence more, actually we have more beans, more MAGs, and we have the more total length of the assembly. But we will find that a majority increase of the total number of MAGs are from low quality, and others, that means it could not satisfy high, medium, and low threshold. So we find that sometimes if we sequence lower and we input more amount of DNA, we have relatively high sequence, larger number of high quality beans and medium quality beans. That means for linked array sequencing, it doesn't mean that sequence more is the best choice. So this is one observation. Another observation for the NFP. So this is all from a simulation data. The different color means the different abundance. That is from the highest to lowest. We find a very interesting things. We find that based on simulation, if we decrease an FP, that means if we decrease the number of the long fragments share the same barcode, we find that the contiguity of the contigs significantly increase actually. So that is also a motivation or next work. So because the 10x technology, Athena designed to 10x technology, so it doesn't consider such kind of the factors. But for the SK-alpha and Tau-seq, they have a lower number of fragments per base or share the same barcode. We can use such kind of feature to increase our assembly quality. Okay, there's some take-home message from this paper. We know C actually total rate depth to demonstrate manner impact on image quality. And for the same C, if we have deeper CR, we sequence more, we got more MAGs. And for the same C, if we deeper CF means that you input more DNA when in your laboratory preparation. You can improve the quality of MAGs, but that is under the same C. Okay, and mu-FF have marginal effect on assembly, of course, because for the metagenome assembly, we know that, or currently commonly only can get 5 kilobase to 10 kilobase fragments. You cannot imagine you have 20 or 30 kilobase fragments. I don't know, but in my experience, it's quite difficult. And the last one is very important. If we find lower NFP, that can lead to better assembly result. Okay, but all of these take-home methods, that's for 10x, and that is for the SINA, this software. But there still have some motivations for our next work. So next work, based on our observations for NFP, so we would like to take the advantage of low NFP in STL-alpha and Tau-seq to develop another method we call the Pangea. We call it using short reads with long range connectivity. So this is from a previous work. Actually, this is for K equals 15 k-mer coverage, because for even we have a single sample, it's quite difficult to evaluate the sequence depth. Okay, because we have the sequence depth, but given 2 contigs, we have 1 contig 15, 1 contig 12. We cannot easily to investigate or determine whether they're from the same bacteria. We have multiple samples. We can calculate the correlation. This is simpler, but for a single sample, it is quite difficult. So here, based on the previous work, we find that if we use a k-mer coverage, we can here easily to separate high abundance, medium abundance, and low abundance context actually. That is good news for us because we have a single sample, we can just share the context into 50 mer. So we calculate the frequency, we can just use such kind of vector to represent the frequency of the context. Okay, and this kind we can replace the 6 read depth as the abundance evaluation, and also we use the TNF. So Panja has actually 2 steps. So this is the first step. So first step, we use deep learning models like the VAE, because VAE have been successfully used to contain binning. So we just borrow ideas there. Here we perform the rate binning, okay, the row rate binning. So we just collect the co-barcoding link rates to larger than 2 kilobase because we want more standard TNF features. So when we collect such kind of rays, we call HXA, so their abundance is 50 mer frequency, and XT is from TNF. We have 2 kind of features. We use this model to perform the dimensionality reduction. We got their embedings. So such kind of things we can get like embedings like this. This from just from mock data, we find that such kind of the rays from the same species can be roughly separated into the different clusters here. Okay, but problem is that this first step 1 works well for high abundance and medium abundance species, because even we have some misclassified the context the rays, that is still fine, because for the high abundance species we have sufficient rays to assemble them, have a high quality context. So this part is designed for high abundance microbiome. But for low abundance microbiome, we have another strategy. We use a name called multi-thread holding reassembly. So we just realign the rays to the context we have, and we can calculate abundance for each context, right? So for the context with higher abundance, we do not consider them in these steps. We only consider the context with relatively low abundance. So actually here we have the different threshold. So we use them to select context, and we collect context and also collect rays aligned to such kind of context, because this, you can imagine this steps looks like we remove from the total rays, we remove all the rays from high and medium abundance bacteria. So we'll only keep the rays from the low abundance bacteria to do the reassembly. We use the multi-threshold to do that. So the motivation of this task is that, so most of the genome assembler, they have very low quality about the low abundance microbes, because when they traverse their assembly graphs, it's quite difficult for them to separate sequencing error and the path from sequencing error and the path from low abundance microbes, because you'll find that they have the quite similar number of rays to support them. So in this strategy, if we remove all the high abundance rays from high abundance species, only the low abundance species, we have a more fair evaluation, okay, to separate low abundance and the sequencing errors on that. Okay, so after that, we merge the context from step 1 to step 2. So just from the mock data, we find that our method got the pretty good like the context continuity, and we compare for tail-seq and STLFR. For the mock data, we believe that you see that the tail-seq gets the best result actually compared with STLFR and 10x, and our method can also better than the previous state of R2s. this all result on CAMI data. CAMI is like a general standard in the field, everybody trusts them. We still find that Pangea get all the statistics get the best result on that. Okay, this is from the real data. So from real data, we find that the Pangea still have the highest number of near complete images will replicate the result in 3 samples, 3 human gut microbiota samples. And also interesting, when we compare the context with the nearest reference genome in the database, we find that our result is pretty good. Even some of them have a circular genomes, and also have a very good consistency with the reference genomes. Okay, so after that, we have the Pangea. We have relatively high quality contigs. As we just said that after we have contigs, we need to group them into MAGs, contig binning. There are many different kind of contig binning tools, but 2 of the previous work, they find that current contig binning tools, there are many contaminated contigs. Contaminated contigs, that is because 2 issues. The first issue is that, so if we use a check-ham to evaluate the quality of MAGs, we are based on the single copy genes. So, but you see that some contigs from different species share the same single copy genes. So they have, sometimes they have such kind of non-redundant contamination, but sometimes they have redundant contaminations on that. But check-ham cannot separate such kind of things. And second thing is that because the contig binning tools are based on TNF, some contigs have similar TNF, but actually they're from different species. If you only have 1 sample on hand, it's quite difficult to separate them. In this paper, they try to align the rates from different places of human body. They find that, for example, the orange contigs, okay, they have the quite different abundance compared with the green contigs. That means this orange contigs is misbinning. So how could we deal with such problem? So then in our groups, so we think the naive way is that we can align the contigs to the available bacterial genomes for NCBI or something else, like string matching problem. But problem is that there are many unknown species. Also, you know, the adjacent species, they may have the like genome arrangement, genomic mutations on that, so make them have a very bad assemblies. So we propose an alternative solutions. We design a method, we use the multimodal genome language model. We want to make a bridge between the taxonomic tree and DNA sequences. That means if you give me a contig, I can place your contig to the phylogenetic tree or taxonomic tree. We do not need the whole genome, and we do not need the whole gene set of the sequences. So how can we do that? So we develop a method named the Purify. So currently you can download them from our GitHub. So in this method, we actually use the contrastive learning, and for each we just train the model, just to share the genome from the reference from bacterial reference genome, and for each sequence we have their taxonomic lineages. This is a positive pairs, and we also generate the negative pairs. We randomly generate the taxonomics, the lineage of these sequences. After that, we perform contrastive learning that to make the model, they can match the bacterial sequences and their taxonomic lineages. We also develop a structure, we call it GS former. This relatively complex, inherited from fish and night, that can efficiently embed the genome sequences. And in this part, we have generating model. So every time you give me a sequence, so we can generate the predominant lineage for the genomes. When we have such kind of predominant lineages, we also have another strategy. We call it separate MAG separate tree. So that can help you separate the genome. Why we design that? Because when you have an MAG, for example, you have 60% of the sequence from bacteria A, 40% from bacteria B. So we do not simply remove like the 30%. We just try to separate them into two images, one from bacteria A, one from bacteria B. Maybe they can satisfy the requirement of CheckM to be high quality beans. So our algorithm in this part is try to maximize the number of high quality and medium quality beans. So based on training set, we compare with the other tools, we find that the performance is great, especially in the, we see the, if they share the core and the contaminated genome, they share the same family and share the genome, share the same genus. Actually, that too is the two commonly phenomenon in your real data. So we also try to perform auto distribution analysis. That means if the reference of genome is unknown, what is the performance of that? Actually, we find that, to be honest, performance decreased a little bit. It's not bad. So same to one here is that the model can see the reference genome, and orange one is our method that reference cannot see the reference genome. So we see that decrease, but the auto distribution performance, I think, is still good. If we find that the contamination rate is not quite high. Okay, this is from some chemi and real data. We apply our method to the different content binding tools. So you can just focus on the last line. We find, for example, for human fecal bacteria, we used 227 samples. We find that for high quality beans, we increase around 30% of them. Okay, and for also for the other more high complexity the community, our method can also increase the high quality and medium quality beans. Okay, this is our demonstration about that. So we find after our method applied to the beaning tools, we find that we will see some actually just images here. Some images have relatively high contaminants, but also have very high contaminations. Your method actually, we just significantly decrease the contaminations of this bean. So we got the more useful images. Okay, so last one, I just roughly introduce our recently developed platform. This platform, we incorporate, so it can support to 10x, STL and TLC technologies. We develop a unified BAM file for the format of BAM file to accept all of them. And it can support both metagenome and human genome assembly, resequencing and assembly. And it can also generate some useful figures for you to investigate your data. And this is some metagenome assembly using IRTK. Actually, in this paper, we use a different real data, but actually we replicate our result findings of a Pangea paper. We find that Pangea is still the best tool and can have much better result than the Athena. Okay, this is, I think, end for my presentation. And last, I will thank for my lab members, my collaborators in US, in Hong Kong, and mainland. Also, especially thank for our collaborators to provide us data from Tom and Ming from TLC and the CFR data from BGI. Okay, so that is all, maybe longer. Okay, thank you.
Thank you very much, Dr. Zhang, for excellent, fantastic talk. Now we are open up for questions. For those who have questions, please feel free to unmute yourself and ask, or you can type your questions in the chat.
Hey, Professor Zhang.
Sure.
Yeah, I have a question. First of all, it's an excellent job. Thank you so much. You know, there's a rich information. You have done a lot of work here. We very appreciate. Yeah. My first question is right. For analysis for the link rates, the the DNA fragment length, the starting DNA fragment is very important, right? Should be the longer,
Right.
the better, right?
Right.
So your result, your information says the DNA fragment only 520 KB, right? That's what you used in the data here.
Actually, this is our simulations on that.
Okay.
Because I didn't find any real data, their mu effects larger than 20 kilobase.
I see. Okay, actually, I just when we do in our in-house and we work with UCSD, Cornell, they all those microbial or metagenomic samples, they can achieve I think 40 KB, 60 KB. The highest we can reach 70 KB. So that's if we got that, that's use your program should get much better result. Expect, correct?
Yeah, I believe so.
Okay, great. Yeah, we'll try to get the DNA fragment as long as possible. Yeah, the tail seeker basically we can handle much longer DNA fragments.
Yeah. If we have
Yeah.
a better DNA extraction protocol, that would be great. And also we also try to another strategy to do the fragment selection. To do the fragment selection, the problem is that we do not prefer such strategy because we need more input DNA if we want to perform the fragment selection.
Yeah, it's the increase the complexity, right?
Yes, right.
Yeah. Another things I saw in your program, you use the you use the abundance information to you tried to resolve the low abundance species. This is very good. I think it actually is a very good feature. A lot of metagenomics researchers that want to know the low abundance one, right? High abundance is very easy to whatever methods to get it. The most difficult one is how to get the lower abundance species accurately identified, right?
Yes,
So,
low abundance. Sorry.
Yeah. I saw you try to remove the reads belong to the high abundant, low abundant,
right.
the medium abundant. So you actually remove some reads, right? You are not use all the reads.
Exactly. That is, for the genome assembly, it's a very common strategy. If you sequence more, for example, even you have 100 gigabytes, the long read sequencing data, you still need to perform random sampling and generate a small set of them to do assembly first. And after that, align the rate to them and remove such kind of rate to do the random sampling again. But link rate, the good thing is that we can, because they use the random sampling, but for link rates we can do the like here we can perform the clustering. We can separate the high abundance species and the low abundance species. Then we can have a direction to remove all the rates from high abundance species, right?
What is the criteria there? How much the risk you keep for the high abundant medium? Any criteria? What is the criteria?
That is a good question. That is also a question from reviewer. Actually, so they ask us how to decide the T here. Actually, the T is like a hyperparameter, so the user can set them. But based on our experiment, commonly we regard if a contig is below 30x, we regard them is maybe a little bit difficult to assemble. So in our paper, so the T we just commonly use 30, 10, and 5, the 3 different abundance strategy. Of course, you can set more, depends on the user.
So the user can have control on that, right?
Yes.
Oh, okay, great. Yeah, follow on is it's amazing. The link rate actually works better than long rates. Get a more high quality. Like max. What is the true reason? Do you think what caused that?
I think, to be honestly, I think there's two factors. Okay, the most important factor is sequencing depth. We should sequencing depth. And second one is that it determines if you use here we use ONT data. So ONT data, for human gut microbiota, I think that is fine. If you apply it to the more complex the environment, you'll find that the Zamli graph is pretty messy. Current method is quite difficult to find a good way to get the high quality context. So I think that is the maybe two reasons.
Okay. Okay, thank you so much. Thank you very much. I'm done.
Thank you.
Okay, anyone else have any questions for Dr. Zhang? All right, so if that's the case, then we are going to conclude this. Let me just stop. And thank everybody for your time. And then also thanks Dr. Zhang for your precious time as well as the excellent presentation. I'm going to stop recording here and thank you. Hope everybody have a good afternoon, good morning, and a good rest of the day and good evening.